Bismillah ar-Rahman ar-Rahim. Assalamu alaikum wa rahmatullahi wa barakatuh to everyone. Today our lecture is about PCR applications. I'm going to divide these lectures into two parts. So let's start with part 1. In this lecture we are going to talk about overview of polymerase chain reaction, which include principles of PCR, components and enzyme of PCR, factors that influencing the sensitivity and specificity of PCR reactions, reverse transcription PCR. Also, we'll talk about methods for detecting PCR products. And of course, important topic is the applications of PCR. Let's do some revision. If you remember about the DNA, we studied the DNA, which is double-strand helix structure sitting inside our nucleus, okay? And this is consisting of nucleotides, or they call it bases. They are complementary to each other, okay? And of course, this DNA will be packed with the chromatin into the chromosome, and it is found in the nucleus. Important term that we are going to hear about it many times, which is base pair. Okay, so this is the base pair, bases of the DNA, that they are complementary to each others. So of course, as you know, the A will be paired with T with double hydrogen bond. Okay, so if it is A present in the one DNA strand, the T will be present in the opposite DNA strand. And if G present in one DNA strand, the C will be complementary to it, and they are going to bind with 3 hydrogen bond. Okay, and this is what we call base pair. And this is the first strand, we call it the sense, and the other one we call it the anti-sense strand. So you can guess, if this is the sequence of the sense strand, what will be the sequence of the anti-sense strand? So imagine the human genome, which consisting of around 3 billion.2 base pair. And from these billions of base pair, you are interested into a certain sequence, which may be few hundred base pairs. So how you are going to find this sequence? It looks like you are looking for a needle in a haystack. So how you are going to find this needle? PCR is a method that will try to help you to find this needle or to find this sequence and to amplify this sequence for you, so that you can proceed with your further study or with your further identification. So what is polymerase chain reaction, or we call it PCR? This is an in vitro method which is used for a rapid production of a very large amount of a specific DNA segments. Okay, I will repeat, this is an in vitro method which is used for the production of a large amount, okay, of a large amount of a specific DNA template or specific DNA segment. What is the principle of PCR? As I said, it is an in vitro replication. In vitro replication of a specific DNA sequence or segment that we are interested in. By this method, we can generate or we can obtain tens of billions of copies of a particular DNA fragment or DNA segment, or we call it the target DNA that we want to do our study on it, or we want to diagnose the disease by searching for it, okay? And of course, this is present in the DNA extract. So we need to extract the DNA, okay? And then we will do the amplification of this DNA by PCR, okay? By looking at the sequence of interest, the sequence of interest that we are looking for it, which is present in that DNA extract, we will amplify it by PCR, and then we will get by this very large amount of this DNA segment or this DNA template that we can look for the disease, or we can do our research, or it depends on what we want to do by it. Why do we need to use PCR? Of course, because DNA is a very small, is a very tiny molecule that cannot be detected, that cannot be seen. So we need to amplify it into a many copies, okay? Into a many copies of DNA that can be detected, that can be searched, that can be looked for, okay? So the DNA is very tiny, you cannot be do anything on it, so you need to amplify it and produce many copies. Okay, keep in mind that PCR is not used to amplify the RNA, and it is not used to amplify the protein. So it is only used to amplify the DNA. Who invented the PCR? In 1983, Carey Bank Mullis invented this process, or this PCR method. So he solved the problem, which is how to make copies of DNA strand that you are interested in it. And in 1993, he got the Nobel Prize in Chemistry. Most of the PCR method can amplify DNA fragment up to 10 kilo base pair. The base pair that we talk about it at the beginning of the lecture, okay? Up to 10 kilo base or kbp. Some method can even amplify up to 40 kilo base, okay? Our haplogenome of human cell contain about 3 billion DNA base pair, which is 3 giga base pair. So let's move on the components of PCR. Okay, for each method or for each experiment, you need few components, right? So you can perform. So what are the components of the PCR? Number one, DNA template. Okay, this is of course the region of the DNA sequence or the target sequences that we are interested on it, that we want to do our research on it, that we are looking for it. Okay, so they call it the target sequence, that we want to amplify it. Okay, the PCR is going to amplify this DNA sequence or this DNA target. Of course, this comes from the DNA extraction. So before PCR, you need to do DNA extraction, get the DNA sample. Okay, then you will amplify it by PCR. So you need to have DNA extraction to get the DNA template, okay, that contains the sequence that you are interested on it. From where we can get the DNA? There are few samples, example, whole blood, okay, the hair root, not the hair, but the hair root, various tissues, bones, cell lines, okay. There are many samples. We can get them, and then we can extract the DNA. Then we can amplify that DNA or that sequence of the DNA or part of the DNA. Of course, we are not going to amplify the whole DNA. We are going to amplify part of that DNA. So how you are going to identify that part of the DNA that you are looking for it? This will be done by primers. By the primers. These are oligonucleotides, okay, that define the sequence, that identify the sequence that you are going to amplify. It may consist of 20 to 30 nucleotides, okay? Identify the sequence that we are looking for it. How? By annealing, okay? We need to have two primers or primer pair. We need to have the forward primer, which will anneal at the antisense. Don't forget that you are dealing with a double helix DNA. So you have the antisense strand and you have sense strand. So the forward primer will come and anneal at the antisense, and the reverse primer will come and anneal at the sense strand. They will anneal at the area. They are designed in a way that they can anneal at the area or the sequence that you are looking for. I will repeat, they are designed or produced in a way that they can anneal, attach, okay, into the area of the DNA that we are interested in it. So you are going to amplify this area according to the primers, okay? Don't forget we have forward primers and we have reverse primer. You cannot do the PCR with only forward or with only reverse primer. You need to have both primers, okay? The next component is DNA polymerase, or they call it Taq polymerase. Of course, this is from its names that the enzyme that catalyzes the PCR reaction. Okay, what does this enzyme do? It extends the new growing strand complementary to the DNA template. What does this mean? Here you have the primer, come and anneal. And this is the other primer as we explained just now. Here comes the role of the Taq polymerase. Okay, Taq polymerase, it will extend the new growing DNA strand. You will have a new strand here and a new strand here. The Taq polymerase will extend this DNA strand. So you will have a new one which is complementary, of course, to the original DNA. So this one will be extended by DNA polymerase, and this one also will be extended by the DNA polymerase. This DNA polymerase will come from bacteria, okay? It is a thermostable polymerase, and of course it is named from thermophilic bacterium Thermus aquaticus. From its name come on. So how this will extend? From where the nucleotide will come. When you extend, you need to add new nucleotides, right? To have something new. You cannot get something new from zero. You need to have something. So the Taq polymerase will add DNTPs, which is called deoxynucleotide triphosphate. They are, of course, they are the usual one, the usual nucleotide adenine, thymine, guanine, and cytosine mixed together in a reagent called DNTPs, deoxynucleotide triphosphate. This is the building blocks of the new DNA strand. Just now I said we will have a new one because we are amplifying the DNA. So you will have a new DNA. From where this new DNA come? It will come from DNTP. Which reagent will add this DNTP? The Taq polymerase. Okay, Taq polymerase will add the new DNTPs, so it will make new DNA strands. Okay, and of course this will be done as the building block will be added, and they will be added complementary to the original DNA. The next component of PCR is the buffer solution. This is containing ion and salts like magnesium or manganese ions. And of course, these are divalent cations that are very important for the Taq polymerase to act. They maintain the pH and the ionic strength so that this Taq polymerase will produce its action properly. This is an example of PCR cocktail or PCR mixture. For each PCR mixture, the genomic DNA and the primers are unique for that PCR reaction. Of course, the DNA sample will be different from each reaction according to the sample that you are looking for. And the primers also will be unique for that reaction because the primer will specify the area or the region of the DNA that you want to amplify. You are not going to amplify the overall DNA, you are amplifying specific area by using the primers. So the primers and the genomic DNA are unique for that PCR reaction. The rest of the reagent, let's say the buffer, the DNTP, and the Taq polymerase are almost fixed for each PCR reaction. Sterile water sometimes we need to put in order to complete the volume into 25 microliter or 20 microliter, it depends. Okay, so sometimes we need to add water. So all those reagents plus the DNA samples will be placed in a PCR tube. We call it microfuge or microcentrifuge tubes. Okay, it's very tiny tube. And then we will place it into a machine. We call it thermocycler. Okay, the microfuge tube will be placed into this thermocycler. And of course, this thermocycler, from its name, it will heat and cool the reactions tube accordingly. Okay. So PCR basically consists of 3 steps. The first step is denaturation, in which there will be a separation of the 2 strands of the DNA from each other's, annealing of the primers, where the primer specify the region of the DNA to be copied, and extension when there is copying process or new synthesis of the DNA. Of course, those 3 steps will be repeated over and over again, maybe 20 to 35 cycles. Each cycle consists of those 3 steps. And then finally, you will get multiple copies of that area or that specific DNA target. So the number of copy is 2 to n, where the n is the number of PCR cycles. So let's have a deeper look on how all these are working. So in the first step, which is the denaturation of the 2 strands of the starting DNA, here there will be separation of the 2 strands of the starting DNA. I mean, there will be a separation of the 2 strands of the target DNA. This will be done by heating the samples up to 94 degrees Celsius, which is almost the boiling point of waters. Such high temperature will cause breaking down of the hydrogen bond between the 2 strands of the DNA. So, of course, the 2 strands of the DNA will be separated from each other. Such denaturation may occur within 30 seconds and sometimes it may take up to 1 minute. The second step of PCR is the primer annealing. In this step, we need 2 short sequences of DNA, which are called primers, that I explained about them at the beginning of the lectures. We have the forward primer, which will stick or anneal at the 1 DNA strand at the beginning of the sequence that we want to amplify. And we have the reverse primer, which will stick or anneal to the other strand at the end of the sequence that we want to amplify. So the forward will stick at the beginning of the sequence and the reverse will stick at the end of the sequence at the other strands. Of course, those strands, they should be complementary to the primer. That's to say, the A will stick to T and the C will stick to G, for example. Okay, and those primers are around 20 to 25 base pair in length. Okay, how we do the annealing? By lowering the temperature. We will lower the temperature. Just now in the denaturation, it was 95 degrees. We will lower it up to 65 degrees Celsius. This will allow the formation of the hydrogen bond to be formed between the primer sequence and the DNA strands. And of course, we will add an extra copies of the primers, so we'll give them chance to stick to the DNA or to anneal to the DNA rather than the DNA will anneal to itself again. The specific annealing temperature is variable from 50 to 65 degrees Celsius. This depends on the length of the sequence and the sequence itself that we want to amplify. And the time sometimes may be very short, 5 seconds for the primers to anneal, and sometimes it may take up to 30 seconds for the primers to anneal. The third step of the PCR is the extension step. So in this step, we need DNA polymerase or Taq polymerase. This enzyme will attach at the end of the region where the DNA and the primers are annealed. Just now we said that the DNA and the primers anneal to each other in the annealing step. So now in the extension step, the DNA polymerase will come and attach at the end of this region, and it will start to extend this sequence by adding the nucleotide. Okay, we'll start to extend the sequence by adding the nucleotide one by one. It will work in the forward direction and in a reverse direction by adding the nucleotides one by one in a complementary manner. So if it finds A on the DNA strand, it will add T. And if the DNA polymerase finds C, for example, it will add G. So by the end of the extension, you will have two new complementary strands. Okay. In order for the Taq polymerase to work, you need to heat up the reaction again into 72 degrees Celsius. Why? Because this is the optimum temperature for the enzyme to work. Sometimes you need 1 minute or few seconds to extend this reaction, or sometimes you need few minutes for the extension step to be completed. So in this slide, I'm just summarizing the steps of the PCR cycle. Denaturation, in which the two DNA strands are separated from each other. You need to heat this reaction up to 94 degrees Celsius so that there will be breaking of the hydrogen bond between the two DNA strands. Then after that, the primers will come and anneal to the two DNA strands. You have the forward and you have the reverse primer. They will anneal according to the specific area of the DNA that you want to amplify. Remember, you are not amplifying the whole genome. You are amplifying a specific area that you are interested in. How you will specify the area? By designing a proper primers. Okay, and after the primers will anneal, there will be an extension steps. Okay, this extension step in which the DNA polymerase will come and it will extend the primer again and again. How? By adding new nucleotides which are complementary to the DNA. Don't forget here the rule of DNTP. From where the polymerase have the new nucleotide, those new nucleotides are from the DNTP that we are adding it in the reaction. So the Taq polymerase will extend the primers and you will end by having a new complementary strand. And don't forget that we need to repeat this cycle again and again, maybe up to 40 times or 30 times, it depends, and you will end by having a PCR product. Okay, this is also another slide. I'm just repeating to make it more clear. Again, here the double helix of the DNA will be denatured under a high temperature. And then the primers will come, the forward and the reverse primer will come and anneal to the complementary DNA strand. And here, by the action of the Taq polymerase with the need of the DNTP, there will be an extension of the strand so that you will end with the two new complementary strands, and the cycle will be repeated again and again, so you will end with the exponential replication of the target DNA that you need. Okay, you have with the millions of copies of the DNA that we are interested in. In order to have a successful PCR, there are certain factors that you need to consider, okay, or certain parameters that may influence your PCR results. Those factors can be physical, like the temperature, for example, the annealing temperature, the extension temperature, and the number of cycles. How many cycles do you need for that PCR to be successful, 20, 30, or up to 40 cycles, it depends. And of course, the chemical factors like the concentration of the DNA template and the concentration of other reagents. So adjustment of all these components of PCR are very important and may alter the quality of that PCR. They can improve or they can diminish the quality or the product of the PCR reaction by affecting the sensitivity and specificity. And because PCR is a very sensitive technique, so you need to do optimization of the PCR before you proceed. By optimization of the amount and the concentration of the reagents or the components of the PCR, including the DNA template and all the rest of the reagents, you may need to be optimized. And also the annealing temperature, melting temperature, all these may need to be optimized before you have appropriate or successful PCR. By the way, all this concentration, no need to memorize at all. Even in the previous slide that I show you, the amount or the quantity of the reagent, you don't need to memorize. However, you need to know the components of PCR. But in terms of the quantity or concentration, you no need to memorize them. Contamination is a very common problem for the PCR. For that, you need to have precautionary steps before you perform your PCR. This done by separating the area of the DNA extraction from the area that you are doing in the PCR. Like, for example, you do the DNA extraction in one lab and the PCR itself in another lab. Don't forget to use gloves, sterile tips also. Of course, you need to clean the benches and your pipettes. Okay, don't forget to add positive and negative control. And of course, you need to carefully measure your reagent and use appropriate cycling condition. Okay, for the positive control, I just want to tell you regarding the positive control here, you are using the PCR template or the nucleic acid or the DNA sample that you are working on it. You put it in the positive control. If you have the PCR product, this will verify the presence of the sample or the diagnosis, like for example, dengue infection. So it will verify that this, yes, your PCR is working. Negative control, there will be no DNA template. You don't put any sample in the negative control. So this is very important because it should not have any PCR product or band. You should not have any band or PCR product. The presence of the band indicates that there is a cross-contamination in your reaction. Okay, so you need positive control to verify that your reaction is working and negative control to exclude contamination of your PCR. Okay, with that, I come to the end of part 1. Thank you so much, and we will proceed with part 2. Thank you. Assalamu alaikum wa rahmatullahi wa barakatuh to everyone. Let's proceed with part 2 of PCR applications. In the first part of the lecture, we talked about PCR principles and we go through the detail of the principle and the steps in terms of denaturation, annealing, and extension. So after we obtain the PCR product, how we are going to visualize this PCR product, how we are going to see and identify the DNA within this PCR product. This will be done through an electrophoresis, which is a technique used to separate the macromolecule like protein and nucleic acid according to their size and charge. When such charged molecules are placed in an electric field, they migrate toward either the positive or negative pole according to their charge. And because nucleic acid are always negatively charged because of their phosphate backbone, they always migrate toward the positive pole. So as I said, electrophoresis is a technique which is used to separate and visualize the molecule like DNA or RNA or protein. Whenever you have an electrophoresis gel, there will be two electrodes. One side is the negative and the other side is the positive electrode. when you apply any electrical power, the negatively side will put its own current, and the positive charge, of course, will apply its own current. Okay, so you will have an electrical current here. So after this, you will have an electrical field will occur throughout this electrophoresis gel. By this electrical field, this will allow the positively molecule to migrate toward the negatively electrode, and the negatively charged molecule will migrate toward the positively charged electrodes, and the neutral molecule will not migrate at all. And because the DNA is negatively charged, as I said, so of course it will migrate toward the positive electrode, from the negative toward the positive electrodes. You can imagine the electrophoresis gel like a sponge with a pore inside it, so that the DNA molecule will migrate through this sponge or through this gel. Of course, the smaller the molecule will migrate faster than the bigger the molecule. You will end with the separation of this DNA molecule, with the shortest migrate faster than the largest molecule, which will migrate slower. We have a few types of gel. We have agarose gel or polyacrylamide gel. Okay, we will make it like it will present usually as a powder, which will be dissolved in a buffer, and then it will be boiled. Okay, and then we will pour it into a cast, and there will be like comb. We put like a comb. Okay, this comb, after solidification, it will produce a wells that we can insert or put the PCR product in it. After that, the gel is immersed in an electrophoresis buffer again, and this will provide ions for the currents, and important to maintain the pH throughout the electrophoresis. So this is how there will be visualization of the PCR product. We will have also a detail in the practical. Here we have the agarose gel as a liquid. We will pour it into the cast and we leave it to solidify. Then we will put the PCR product in the wells, and then we apply an electrical current, and then we will visualize it in a gel dock to look for the band. After the electrophoresis, the gel that contains the PCR product or the DNA fragment will be stained usually with the DNA dye. It can be ethidium bromide or cyanide green, and then it will be viewed under the UV light, okay, in order to be seen and analyzed. So this is how it is look like after visualization. Here we have a leather which contain a known size of the DNA. This will act as a ruler for us, to rely on it. So as I said, the smaller the DNA fragment, the faster the migration. The smaller the DNA, the faster the migration. And here in each area of the gel in which a single sample of the DNA is migrate, we call it a lane. So this is the first lane, second, third, and fourth lane. And as we can see here, more detail, this is 100 base pair because the smaller, so it migrates faster, 200, 300, and 500, and so on. Okay, and this is the first lane. There's two bands. This is we call band. This is the size of the band. It is between 100 and 200. So it's estimated 150 base pair. Here between 200 and 300, estimated 250 base pair. And here the second lane, you can see the size of the band. It is almost 400 base pair. This is how we are looking and do the interpretation of the PCR product. The most common types of gel that are used in electrophoresis are agarose gel, which has a very large range of separation. And the other one is polyacrylamide gel, used for separation of the fragment which is less than 500 base pair. We have a few types of PCR. The one that we talk about it is the conventional PCR. We have another types like reverse transcription PCR, RT-PCR, multiplex PCR, nested PCR, and the real-time PCR, or they call it qPCR or quantitative PCR, and the other one is single-cell PCR. The first one is reverse transcription PCR or RT-PCR. RNA cannot be used directly as a template for the PCR. Or in other words, PCR cannot be performed on RNA because the DNA polymerase or Taq polymerase cannot recognize the RNA as a template. Don't forget that we have the DNA as a template. Here we have RNA as a template. RNA cannot be recognized by the DNA polymerase. So in this case, we need a reverse transcriptase enzyme or RT enzyme. This will use RNA as a template to be converted into the DNA strand. So there will be a synthesis of complementary DNA by incubation of the RNA with the reverse transcriptase in the presence of dNTP and primer. And you will have the product, which is the complementary DNA, which will be used directly for the PCR. So in summary, you need to convert the RNA into complementary DNA by reverse transcriptase enzyme with the presence of the dNTP and primer to have complementary DNA to be used for the PCR reaction. The second one is a multiplex PCR. In this type of PCR, there will be a simultaneous amplification of many target sequences of interest. In the conventional PCR, you have only one target sequence or only one target DNA. Here, you will have a multiple target or multiple DNA, which will be amplified. And of course, in this case, you need more than one pair of primers. Okay, so more than one pair of primers with more than one sequence or DNA sequence will be included into a PCR master mix and in the thermocycler, so you will have a multiplex PCR product. The other type is called nested PCR. This type of PCR is used to improve the sensitivity and specificity of the PCR. Here we are using two sets of primer. The first round of PCR or the first PCR reaction, you use one set of primer which is called an external primer pair, and the second PCR we will use the internal primer pairs, which is designed to amplify region within the first set of primer. Okay, so this is also to explain about it. Here the first PCR external primer, then the second PCR internal primer, which is within the region of the external primer. If there is no amplification, this will suggest non-specific amplification of the PCR product or of the first PCR reaction. If there is an amplification, this will confirm that there is a specific reaction, okay, or there is specific locus to be amplified. So as I said, it is used to confirm the sensitivity and specificity of the amplified PCR product. Single cell PCR, this will amplify the total DNA from a single cell. And usually this is used for the diagnostic, like for example bacterial, like for E. coli, because it is work on a single cell. The other type is called real-time PCR. This type of PCR is used to amplify and at the same time quantify the DNA molecule within that PCR product. It is quantitative, so sometimes they call it quantitative PCR or qPCR. It's used to quantify the amount of the genome in that sample. How? By using a fluorescent dye. This fluorescent dye which labels the PCR product during the cycle. Example, Tatman probe. This is target-specific oligonucleotide that produce a fluorescent signal when the target DNA is being amplified during the PCR reaction. The second one is cybergreen dye, which will bind to a double-strand DNA and emit fluorescence only when it bound to it. This real-time PCR measures the accumulation of the fluorescent signals during the exponential phase of the reaction. It is very fast and precise quantification of the PCR product. The data are usually collected in a real-time PCR reaction will proceed, and it does not require detecting PCR product by gel electrophoresis. So it does not need gel electrophoresis because it is using certain software. Real-time PCR, as we said, monitor the fluorescence which is emitted during the reaction and use it as an indicator of the amplicon production at each PCR cycle in real time. For that, we call it real time as opposed to the endpoint detection. So what are the applications of PCR? Each technique has its own application. So in terms of PCR, it is important for diagnosis of infectious diseases, detection of hereditary or genetic diseases, DNA fingerprinting, or they call it DNA profiling, cloning, paternity tested, and a lot more. For the detection of the pathogen, you will be provided with a sample that may or may not contain pathogenic DNA, like for example, dengue virus, coronavirus, and so on. You will perform the PCR, okay? And if you get the PCR product, you mean that you have the pathogen, okay? Or there is pathogen in that sample, or the diagnosis is positive. If there is no PCR product, what does this mean? This means that there is no pathogen, or the sample is negative for that pathogen. So example for detection of pathogens by using a real-time PCR application, quantitative measurement of HIV virus copy, and detection quantitation of Plasmodium falciparum DNA. Another important application of PCR is the detection of hereditary diseases or genetic diseases when the DNA is extracted from chorionic villus sample or amniotic fluid or from blood, okay, where the specific primer will be designed according to the sequence or the mutation that we are looking for in certain gene. And many diseases will be diagnosed by this PCR technique, like sickle cell anemia, thalassemia, familial hypercholesterolemia, and many more. Another important application of PCR is DNA fingerprinting, or they call it DNA profiling. This is used to identify or evaluate the genetic information or the DNA of an individual. Here it is used in the forensic lab, where a non-coding area which contains segments called short tandem repeat, or STR. This is very short DNA sequence that is repeated from as a head to tail fashion. Okay, the number of this repeat or STR marker is highly variable among individuals, or it's very variable from one person to another. So this region or this STR is used as a marker for the human identification purpose. This is example of DNA fingerprinting in crime scenes, where you have the victim. This is the DNA profile of the victim, and then you have a few suspects, 1, 2, 3, 4 suspects. So you need to compare which one is matching with the victim or with the evidence from the victim. And here, of course, it's very clearly that suspect number 3 is matching with the evidence or with the victim. Okay, so we can assume that this is the guilty person. And other one also is used for paternity testing or for ancestry DNA, when you have an adopted child, for example, and you have the mother, and for paternity testing. Okay, so again, you can match. You can match here and look at the suspected father. Okay, which one is more matching with the child? Okay, we can identify that the male number 1 is more matching with the child than male number 2. So this is also another application of PCR, paternity testing and ancestry DNA. Another important application of PCR is cloning of the gene. Okay, here in this process you have the vector, okay, and with the insert. Vector and the insert are digested together with the restriction in the enzyme. And of course, the digested fragment will be mixed together and ligated. Then you will have a recombinant plasmid, which will be transferred into a host, like example, the E. coli, and then this recombinant plasmid will be propagated into the host prior to screening, and then this will be transformant will be selected on a plate which containing appropriate antibiotic. Okay, this is a simple overview for gene cloning. So here is some references that you can refer to for my lecture. And of course, I need to acknowledge Associate Prof. Dr. Wang Somoy and Associate Prof. Dr. Mudiana. Here are some of the links for YouTubes that's really helpful and really interested and very simple, that will help you also to understand my lecture. With that, we will end with part 2 of PCR applications. If you have any question, you can contact me, and also we can discuss in the MS team and during the SGS. Thank you so much for listening.